BCG Substrains, myths and fact. Well, in the COVID-19 era, we are all talking about and strains. Today, I'm also speaking to you about a strain, but a different strain, that is BCG Substrains. Why we are talking about it now? Why this confusion even after 45 years of introduction of BCG vaccine? Well, Mycobacterium bovis bacilli calmette was introduced after 230 recultures of a pathogen known as Mycobacterium bovis, and it was cultured over 13 years to make it non-virulent. And 2021 marks the centenary year of BCG vaccine. Morals in the year 1976 introduced its use in Bladder, and later Lame et al. Confirmed the finding in 1980. What we have understood from BCG vaccine for tuberculosis is there are 6 substrains of BCG which are approved by WHO, and all of them have got fair immune response. There's no obvious variation in immune response, and there's no particular strain which is superior to other. And this is well published by Rist et al. In a review literature. There are different types of Mycobacterium available, and they are different from substrains what we talk about. The Mycobacterium are basically divided into cultivable and non-cultivable, and basically typical and atypical based on their growth patterns and the pigmentation. But we are not going to talk about this form today. So how a vaccine actually becomes avirulent? Common to all BCG vaccine strain is deletion of RD1, which is preserved in the virulent form of Mycobacterium bovis and tuberculosis. Loss of this region is known to have been critical event in attenuation of initial Mycobacterium bovis strain. But how substrain came into existence? Well, once the BCG was produced by initially, BCG was sent to different countries and it was recultured in local lab according to their own protocol for maintenance and production, which have led to different substrains. So substrains were basically divided into early and late substrains. First, the elimination of RD1 resulted in the early strain, which accounts for BCG Russia, Morio, Japan, Sweden, and Brickhug. Later, the RD2 deletion resulted in Prague, Galaxo, Danish, Thais, Canard, Philips, and Pasteur. Basically, probably many studies actually quote that probably the early strains are better immunogenic as compared to the later one. So what changes in substrain? Basically, deletion of some regions of their genomes or inclusion of single nucleotide polymorphism or insertions of sequences or appearance of tandem duplication. But I'm not going to go into detail of each and everything. And it is published by Keller et al. In BMC. Well, all these changes lead to cell wall changes which are highly hydrophobic and impermeable, not equally present in all substance, which results in different response to immune system, lipid immune receptors. The immunogenic and safety efficacy of different substances is also being researched now. So what is the reason for discussion today? Well, the most paramount importance which have led to this discussion is vaccine shortage. The uncertainties, same clinical efficacy and effectiveness still persists. Comparison of different strains become difficult because different strains are being used in different studies, and the adverse effect profile is still unknown of different substances. So, what is the beginning of this problem? Actually, all this problem started when Sanofi actually, during a routine protocol checkup by FD in the Sanofi culture factory, they found mold there. And this led to the stoppage of the production. But in year 2017, they existed from the market, and this led to shortage of the supply. Sanofi was the major supply in North America, Europe, and several places in the globe, and resulting in supply constraint. So can we combine strain? Of course, we can combine strain, but the data are not very prominent or conclusive. BCG Canada and BCG Japan were compared. The switch of substance during the treatment reduced the adverse effect found at the beginning of the treatment, which was said by Niva et al. But there was no difference in progression-free survival was found in a study conducted by Rentish. Of course, they said the recurrence-free survival was better in BCG Canat as compared to your BCG ties, but the progression-free survival was almost same. They also found there are several studies which also found similar recurrence-free survival of different substrains. The studies published by Guru et al., Uda Urezi, Karajaseki, and D. Andre, all of them have said that the recurrence-free survival is similar as compared to other vaccines. So are the other vaccine or other strains safe? Yes, of course. In a study published in Canadian Journal of Urology by Tan et al. Have clearly said that there's no superiority of one strain over the other, and during this supply constraint we can use any vaccine for our patient. What about the efficacy of the vaccine? Well, in a meta-analysis comparing 10 different substrains, they were unable to find the best strain. And mind you, we have also seen the BCG vaccine, which is given for tuberculosis, who have already recommended six different substrains of vaccine. What about the EUA guideline? They have clearly said that there's no one clear substance which is superior to another. So whatever the strain which is available in your local market can be used. So there are many other parameters other than strain which account for BCG vaccine efficacy in bladder cancer. So to conclude, variability of BCG strain is not demonstrated to affect clinical outcome in bladder cancer or tuberculosis vaccination. The mechanism of action of BCG vaccine in bladder cancer complex and involving many subcomponents which individually can stimulate a robust, adequate immune response. Many other parameters other than BCG strain accounts for clinical efficacy and safety of BCG. And at the time of BCG shortage, difference in strains are not of major concern. Thank you.